This is the Viral Genome Annotation workshop webinar. Just wanted to introduce you a little bit to some new things that are happening at the BV-BRC. So please follow us on LinkedIn if you aren't already. You can use this QR code to do that. I'd love to invite you to the next workshop. So this is a 5-part series. Next time we'll be talking about how to build phylogenetic trees in the BV-BRC. And I do have April from my team here to help answer any questions. So if you see her responding to anything, she's another BV-BRC person. I work on the BV-BRC project on our viral team. I work at the J. Craig Venture Institute. At the end, I'll talk about the institutes that are coming together for the BV-BRC. Last time when I did this, I didn't say who I was, so I just wanted to tell you all who I am. I'm a master's from the Keck Graduate Institute, and I did my undergraduate at RIT, and I spent some time at Uppsala University. Just a quick background on me before we jump into the topic. Similarly, I wanted to just run through what the BV-BRC is. So if you're not familiar, we're a one-stop shop for data about human pathogens and related bacteria and viruses. And we have a resource that has a bunch of bioinformatic tools as well as a robust database that has the genomic data for those pathogens. And thinking about our tools, we have a bunch of different tools for analysis. So I just wanted to go over some of the tools that you can use for annotation today. Broadly, some key features of the BV-BRC is that we ingest data daily. So we get data from GenBank that we pull the genomic data from every day. And they are annotated internally. And we do some manual and automated curation of that data to make it more searchable and usable for your research. We also use different tools like phylogenetic trees, the workspaces that you have private to you to do your analysis, API and command line tools, and then outreach and education like this. So if you have a robust BV-BRC community at your organization or in the geographical area near you, that's one of the ways that we host workshops, and definitely connect afterwards if that's something that you're interested in. But today I'm going to tell you about a few of our services and tools. So spend some time talking about the FASTQ utilities, which are for aligning, measuring base quality, and trimming FASTQ read files. Our genome annotation tool, which uses the Vigor in the backend. And our SARS-CoV-2 genome analysis service, that's like a meta-analysis that takes raw reads to assembly annotation and variant analysis. So my objective for this conversation is to describe those BV-BRC tools, give you an understanding of the annotation algorithms that we use, show you how to upload reads or contigs and submitting a job in the BV-BRC, and then viewing and interpreting those results. So when we're thinking about which tool to use, depending on where you start, the tool will change for the file type and the goal of your analysis. So that's when it's really important to come to the BV-BRC with a question, and that will guide what data you're using and then subsequently what tool you might want to use for your analysis. So similar to studying a specific virus, the tool that you use will impact your analysis. So our FASTQ utility service makes common FASTQ file operations accessible. So you can generate FASTQ reports on base call quality, align reads to genome, which we use Bowtie to generate BAM files, save unmapped reads and generate SAM stat reports, trim adapters and low quality sequences using trim galore and cutadapt. Can also use them to define a pipeline of activities. So we have 3 components: trim, FastQC, and align, and those components can be used interchangeably in whatever order you might want. So you might want to align, then trim that are in the FastQC, or use FastQC to see which reads to take further, then trim, then align. That pipeline plug and play adds a lot of versatility depending on your analysis. And we'll go over that more when we're actually looking at the resource, but that is one benefit of the FASTQ utilities. And you can use paired or single end, long or short, zipped or not, or an SRA, a session number in the FASTQ utilities service. Thinking about our genome annotation and assembly, we support the SARS-CoV-2 virus for that. So this service is the meta service that accepts raw reads and performs assembly annotation and variant analysis. And our annotation service uses Vigor4 and accepts FASTA formatted config files. And then based on the taxonomy of the associated virus, you get an annotated genome and this viral genome ORF reader. So I just wanted to go a little bit deeper into the background in some of the tools. So we'll start with the SARS-CoV-2 service. Thinking about assembly, those are the sequences produced after chromosomes have been fragmented and then putting them back together. So we know that sequencing technology can't read whole genomes in one go, but has small pieces. And some viruses have RNA genomes that can be reverse transcribed into DNA before they're sequenced. So our SARS-CoV-2 service takes in either your submitted FASTQ files, and here's a good time to stop and think about how data can enter the BV-BRC. So you can use our database and pull data, or you can use the SRA accession numbers, or you can have your own data from your sequencing projects that you load up in a private workspace. We never see it. It's kind of like a Google Drive folder, and that was something that we talked about in the last webinar. So once those links are live, you can review that or any of the help documentation about how to make a workspace if you're unfamiliar. But however you bring in the FASTQ files, depending on where they came from, so if it was sequenced on an Illumina machine or a nanopore, the trimming, mapping, and variant calling might be a little bit different. This is also all in our help documents. Then you end up with a consensus FASTA that's run through Vigor and then set up so you can submit it to GISAID or GenBank after this process. So that's the basis on the assembly. Then the annotation side is the Vigor form, which was developed at JCVI, and it predicts protein sequences encoded in a viral genome by sequence similarity searches against our curated viral protein databases. You can see those steps here. And when we're thinking about analysis of the SARS-CoV-2 service genomes, you could have paired read libraries, which is you loading that data yourself, single read libraries, or the SRA session number. So I was preemptive and I already had a slide about this. So with FASTQ utilities, similarly, you will have parameters that you can put in just of where it's going to end up, or those pipeline steps that I told you about, but we'll look at those a little bit closer on the site. Just wanted to show you the FASTQ utilities service quickly. When you're thinking about inputting paired or single reads, it's important that you have those files on your machine that you want to use for this tool, or already have it in your workspace, but it wouldn't be necessary if you had an SRA number. Just had these slides to show you how to do it, but I will show you once we go through the tool. So thinking about the annotation tool, the SARS-CoV-2 service is multi-step. The annotation tool takes in a FASTA formatted config file and then the annotation recipe, specify the taxon of interest, and then runs annotation. So this is just going to be annotation, not annotation, assembly, and variant calling. The viruses that we support with Vigor4 are listed below. And so if you're working on any of these viruses, you are able to use the viral annotation tool on the BV-BRC to do your analysis. So you can see this long list of viruses. And this is posted on the GitHub. Here are some of the components of Vigor. So thinking about ribosomal slippage, shared CDS, and functional length, max gene overlap, alignment score factor. These are some of the things that you'll be able to get out of those annotation reports. But now I think is a good time to jump to the research itself and begin to play around with these tools. This is the homepage of the BV-BRC if you're not familiar. All of our tools and services exist under this tab. We will start with the FASTQ utilities, which is under the utilities, so in the bottom right corner. Feel free to follow along with me on your device. The first step, I will be using a file on my local machine, so that won't be follow along, but for the SARS-CoV-2 genome analysis, we'll be using an SRA number, so you can follow along with that. So each of our services have this similar structure of little gray boxes, and can see the quick reference guide, tutorial, and instructional videos. If we click on the quick reference guide, this is just gives you a quick overlay to be able to run a job with that tool. The tutorial is a lot longer, and so if you're teaching a class or if you're just trying to understand how to use this tool past our meeting today, this would be a great resource for that. And then we also have instructional videos on our YouTube. So that's a great way to check out information about the resource and get that step by step. And these videos will be posted there afterwards. So going back to the page, the parameters are going to be where you want this job to end up. So each workspace comes loaded with a genome group, a feature group, and an experiment group. I'm going to put this job in our experiment group. Zika FASTQ today's webinar, but you can name it whatever you like. I highly recommend being as organized as you can, but just for show, that's what we'll do now. So the pipeline, this is where I was talking about the order. So you could trim. So depending on whatever you want to do, you put the workflow of choice. So we'll start by aligning, and then you must press this little plus to get it to be run as a step. Then we'll follow that with a trim. Now you can see that aligning is on top and then trimming. So thinking about first we'll align, then we'll trim, then we'll run a FASTQ report. And since this is actually, I will do a SARS-CoV-2 one just so we can use an SRA number. So here, just start typing. Sometimes it's a little finicky. So you can scroll all the way down. That's not always the easiest. So gonna have our target genome be this severe acute respiratory syndrome coronavirus 2 strain. And this is just an example, but could use one of these other ones depending on your research. So I'm going to stick in the SRA number since I have one of it handy. And you will need to move it over so that it can run that job on that. So it will do a validation step to make sure that this is actually an SRA number. And then that's loaded and ready to run the job. Now here you could, if you had paired reads from your sequencing work or a single read, depending on the machine, we use different recipes, but just so that if you wanted to follow along, you could use this SRA number. So I'll give everybody a second if they wanted to follow along to load up the SRA. But that's all you need to do to get a job set up. So now this job is loaded and ready to go. I'm going to hit submit. Now you can see that it's successfully been submitted, and all of your jobs exist over in this bottom right corner, so you can see them running. The FASTQ utilities is pretty quick, so probably run as we're doing this once it's started. But I'm gonna go back to our next tool so that we can show you how to start a job in the annotation tool. So I have loaded up a Zika virus genome that I got from this accession number in CBI. And even though it says .txt, it's in FASTA format. That's just how it downloaded on my computer. That kind of shows how smart the BV-BRC uploading system is. So if I hadn't already uploaded it, just going to select files. And because this file is actually formatted like a FASTA, but it's saved as a text file, you might not be able to have access to it at first. So then you just remove this restriction And by removing that restriction, you're able to upload any of the file types. Now, obviously, my QR code isn't going to give me a FASTA file, but I know that this is FASTA formatted data. So even though my Mac has said it's a text file, the BV-BRC doesn't care about what that extension is, just that it's formatted correctly. I wouldn't play around with that too much, but it has to be formatted as a FASTA file with the way that a FASTA file is structured. So then you can upload that, but I've already uploaded this, so I'm just going to click on it in my workspace, and I'm just going to start over on how to select it. So you can either pull up your recent files, which is what this is, or you can go to a specific folder. And so I just stored this in my genome groups. So then I'm going to hit OK. This show all files and folders. It will pull up anything else that might be there that's hidden because it's not relevant. So if you felt like there was something else that was supposed to be there, then clicking on that will help you find that. I'm going to hit OK. For the annotation recipe, we're going to do viruses, and this will tell it to use Vigor. Again, the same quick reference guide and tutorial are here to use. And I told you that this was a Zika virus, so I'm going to use the species Zika virus, and then I'm going to label it with the accession number, but this is just for myself, and I'm going to put that in the experiment script. So that will be the output of my file. And now I'm going to annotate this genome, and we'll see what we've got. So similarly to FASTQ utilities, the job will load. Sometimes it takes a second to show up on the job status page. So I just reset my screen, and now you'll see that that annotation job is running too. And there might be a bunch of stuff happening right now, so I've run these already. So when it comes time to look at the reports, you can see that, just in case it's taking a while, but that FASTQ utility is already running. So the final tool that I'm going to show you how to submit a job is the SARS-CoV-2 genome analysis. And we're going to use that SRA number from before. So we're going to start with read files with the accession number of 11607707. Just move that over into the selected libraries. It does that validation step. And you can do a paired read library or single read library if you're running SARS-CoV-2 sequencing on your own, which is definitely a great functionality of the BV-BRC. This is just having something that someone else has done for the purposes of sharing with you all. So there are a few different parameters, and this is back through those different methods that we talked about at the beginning. Auto is a great option. You can also choose what primers you would like to use, depending on what you're starting with. I'm just going to leave everything as it is for now, and I'm going to label it based on the accession number, and I'm going to put it in the experiment. We'll give it 2 since I've already done that. So this is kind of getting us ready for this job. So if you want further information about the parameters, see about what strategy to use, Auto will tell you based on reads provided. CDC Illumina or Nanopore were original CDC tools from the beginning of the pandemic. And the Arctic Nanopore uses the Arctic prescribed protocol, and the taxonomy name is pre-populated, taxon ID pre-populated, your label we just talked about. But there are always help texts and things like that along with the tutorials and quick reference guides for you to use. So definitely use those to help with your analysis. So now we're ready to submit. Let's go submit that job. And that has successfully been submitted, so you can see that running. And now I'm going to start going through the outputs of the jobs. So the FASTQ utilities job that I ran this morning had, and see what sort of comes out of that job. Let me back up for a second. So each time you run a job, have an output depending on what the job was. So with the FASTQ utilities service, depending on if you did annotation, aligning, trimming, in what order, you'll get different files as an output. But a good place to start with any job is the view. And so this will automatically bring you to the FASTQ utilities report. And the eye is kind of guiding you where to go based on what's most commonly looked at for those tools. But you have access to all of the different files, so if you could use those for further analysis if you needed. But you can also click on the report. So here's that report as a text. So we use the paired end trim galore version 0.6.5, cut adapt 4.2. So this is all good information if you're publishing it or want to replicate your work. There were 477,000 reads processed. These are the criteria that we used. They're the sequences that were removed. This is just in text form, not the easiest to look at. Sometimes it's hard to go back to the view you're expecting. This time it's fine, but you can always go back to the jobs page and click on the job that you're interested in, double click, and that'll bring you to the job overview. So you can view the trim, align, or FASTQ. So I'm going to do the trim report. That's what we were just looking at. Could do the align report. This is an HTML view. You get the mapping qualities, read length distribution, base composition. These are all figures that you can change the view, download, use for your papers if you're interested. Mismatch distribution. So this was the align. Then you can do the QC report, showing quality, GC count. Just depending on what you're interested in, you have these to use. And again, the different files, unzipped or zipped. So that's the standard output for a FASTQ utility. And the exact information would be different depending on the order you took those steps in. Glad to introduce you to that. Then thinking about the annotation tool, I'm going to click on that job. This genome had a bunch of features associated with it. To click view and see it's made a genome view for this sequence. And sometimes this takes a couple hours, a few minutes to load. So if this doesn't come up again, I'd check back in a few hours and see if you've got it. So that it's one coding sequence, 10,000 nucleotides, and GC count of 51. And view the genome. So that also takes a minute to load. The annotated features. So there was the whole genome, which is to 10,272, and then some proteins that were annotated to merge your peptide, an envelope protein, and non-structural proteins, so on. And depending on the virus that you're working on, you would get different things annotated. Those are the features, but they're also annotated as proteins. If there was an associated PDB structure for anything, that would come up here. This is just a view, like the database view that you would get if you were looking at a BV-BRC sequence on its own, but just internal for you. And then it will pull up some publications associated with Zika, but again, this is a private view for you. So I'm going to go back to the jobs page and show you some of the other files that we have here. So you have a quality report as a JSON file, the standard vigor outputs, and if there were any warnings, so something that was unexpected, then the features as a FASTA, proteins as a FASTA, the original file that you started with, the .genome.gdm. So all of these different files are a great place to, depending on where you're going to take it next, you have access to all of these files along with the different views. So that's the overview on the annotation tool. The comprehensive SARS-CoV-2 analysis. look at next. I just want to show you quickly. Here you can see that I have a successful completed job with this SRA accession number, and this is actually a bacteria. So it will run the service if you're using something that isn't a SARS-CoV-2 SRA number, which is just something to be careful about, just to not waste your time, but nothing comes out of that. So just a reminder that if you're going to use an SRA number, make sure that you're starting with a SARS-CoV-2 genome. But this one is actually SARS-CoV-2 genome, so we can look at the output of that. One thing that I haven't shown you on any of these views is the parameters. So this is just a way for you, in a very codey way, seeing what you put in. If you're trying to remember what you did someday and you want to be able to replicate it or change something, this is a good way to know that I put Arctic primers and viruses as the domain, and just kind of those things to make the job reproducible. You can also rerun a job. On the second Thursday of the month, we will have scheduled downtime, but maybe you forget that we have scheduled downtime and you start a job and then that job is canceled because the site is currently down. The rerun feature would be a great way to just start that job over, just so you can run that analysis. But say with this failed job, I'm not sure why it failed. You go into it, the object isn't found. But I had an object there. I don't understand. So then you can click on the report issue, and that'll give our team the information about what happened to the job, why it failed, and it'll be associated with your BV-BRC ID. Then we're able to follow up with you to get you any information that you might need and fix that problem. So that's a great tool to use, just a plugin for a report an issue. And then you can also view things there. But this green bar is pretty reproducible on all our tabs, just has different information. So always check it to make sure that you are aware of what's happening with the specific tab. So it's going to look different on the job status as it's going to look on the job report. But now let's look at the SARS-CoV-2 analysis. So here you can see there are different things on the green bar, but it's still there. So the eye is going to bring you to the full genome report. We'll click on the eye. So we've got some information with the pango lineage, SNPs that were assembled, genes, what we input for the assembling, the coverage. Again, you can always use these figures in your reports. Just make sure you're setting the resource. Variants that were found in this genome. So at position 8,782, there was a variant that changed the codon, for example. We annotated ORF1A polyprotein, SP2, and so on. These features are all annotated on that genome, so for helicase and so on. Then just some information about the SRA session, so the paired read, Illumina miniseq, used the Arctic protocol, and that's why we kept Arctic, or we used auto, and then it matched that. So the succession information isn't going to come up if you didn't use no succession one. Ambiguity score, lineage, QC status, and so on. So this is just the whole report with a lot of different information that you can use. Then there's also talked about how this is both an annotation and assembly tool. So you can click on within the job the specific annotation, which has all of those annotation job we ran. It's very similar files. You can view those in the same way. And then on the assembly side, this is new. You can see JSON files, and FASTA assembly detail PNG. It's just an image for the cover duct. In this case, it wasn't very different across the places. And this is where the SRA metadata is also stored. So you can always click through these and download them or use them within the BV-BRC depending on what you're interested in. So I just railroaded you all with a bunch of information. So just to recap everything that we talked about what is the BV-BRC, gave you a description of 3 BV-BRC tools, showed you how to run jobs, and then we very quickly went through the output of those jobs. Again, there's way more specific information about all of this in the tutorials. I just wanted to show you all a high-level explanation. I also mentioned at the beginning that the BV-BRC is a multi-site project, so we have people at the University of Chicago, JCVI, University of Virginia, UAB, and the Fellowship for Interpretation Genomes, and we are a NIAID-funded bioinformatic resource. Thank you so much for listening. Does anybody have any questions? Mutations is something that we are going to be annotating just so the recording understands. The question was, is there a way to check for viral mutations? We have some tools that are going to be mutation specific across viruses that are in the process of being developed. We can also align a bunch of sequences and find variants that way using the multi-sequence alignment tools that we have in the meantime. It's not like annotated mutations, which is something that is good. If you have any questions after this, we're always available at help@bvbrc.org. So feel free to email that or can reach out through the contact us on the site. So if you also have any questions at any point, just go to about, contact us, and this will immediately send you back to our team. You can just say, help with annotation, and then put in your message, and then that will submit it to our team and we'll be able to answer anything that you have. Thank you all for coming and have a great rest of your day.